When salt moves from the filtrate to the interstitial fluid, water follows by osmosis. The salt and water then diffuse into the peritubular capillaries. Glucose and other essential substances are also transported to the interstitial fluid from the filtrate, eventually going into the peritubular capillaries. Cells of the transport epithelium secrete H+ into the lumen, synthesizing and secreting ammonia at the same time in order to trap H+ in the form of ammonium ions. The proximal tubules also reabsorb about 90% of the buffer bicarbonate from the filtrate. Many wastes leave the body fluids during a non-selective filtration process and remain in the filtrate while water and salts are reabsorbed.
Finally, reabsorption of water continues as the filtrate moves into the descending limb of the loop assembly. Here, numerous water channels formed by aquaporin proteins make the transport epithelium freely permeable to water. For water to move out of the tubule by osmosis, the interstitial fluid bathing the tubule must be hyperosmotic to the filtrate. This condition is met along the entire length of the descending limb. As a result, the filtrate loses water and therefore its solute concentration increases all along its journey down the descending limb.
What happens is that the filtrate reaches the tip of the loop and then travels within the ascending limb as it returns to the cortex. The ascending region has two specialized regions. One of them being a thin segment near loop tip, and then the other one is a thick segment adjacent to distal tubule. As the filtrate ascend in thin segment, sodium chloride diffuses out of permeable tubule into interstitial fluid, and then in the thick segment, movement of sodium chloride continues.
Tube. The distal tubule plays a key role in regulating the K+ and NaCl concentration of body fluids. This regulation involves the variation in the amount of K+ secreted into the filtrate as well as the amount of NaCl reabsorbed from the filtrate. As the collecting duct traverses the gradient of osmolarity in the kidney, the filtrate becomes increasingly concentrated, losing more and more water by osmosis to the hypersomatic interstitial fluid. Because of the high urea concentration in the filtrate at this point, some urea diffuses out of the duct and into the interstitial fluid. Along with NaCl, the urea contributes to the high osmolarity of the interstitial fluid in the medulla. In producing dilute rather than concentrated urine, the kidney actively reabsorbs salt without allowing water to follow by osmosis.